slide, it just has a quick summary of, hey, here's what, could you please turn these lights off for a bit? Thank you. Just the first one, that's good. This slide is basically talking about what the key points that we need to learn. And so we're going to talk about mono, di, and polysaccharides. Saccharides are sugars. And yes, we are starting biochemistry with chapter 16. Biochemistry is going to cover 4 major areas. Carbohydrates, chapter 16. Proteins, chapter 18. Lipids, chapter 17. Lipids are, you're going to find these pretty organized. The saccharides have a common structure. The amino acids that make up protein molecules have a common structure. What do chapter 17 have in common? They're not soluble in water. That's what lipids are. What do they have in common? Well, you're going to see they're a pretty diverse group. There's a pretty diverse group of molecules that we would call lipids. There are triglycerides, fatty acids, steroids. There's phosphoglycerides. There's just a bunch of things. So we'll try to show as many similarities and then the distinguishing difference between some of these. But the main similarity is they're not soluble in water. Amino acids are soluble in water. Sugars are soluble in water. You take a teaspoon of sugar, gone. It's soluble. Dissolves in your coffee. All right? It's floating around in your bloodstream. It's very soluble in the aqueous environment of your bloodstream. But lipids are not. They're not that soluble. They're stored. They're transported and things like that. Okay, so carbohydrates, proteins, lipids, and then the fourth one is DNA, RNA, nucleic acids. These are the 4 main categories, groups of biochemical molecules. And we're in here on the first one, carbohydrates. And what makes up carbohydrates are monosaccharides. So that's what we need to get familiar with first, is the individual monomer, the individual molecule that is a sugar. What does that look like? And then how do they link together to form disaccharides, and how do they link together on from there to form polysaccharides? All right. Know what a chiral carbon is. This has to do with optical isomers. This is a kind of isomer you haven't been exposed to yet, at least not in here. Optical isomers. And I'd like you to appreciate what it means that there are optical isomers. This is where living systems really, really shine, because in the laboratory we can't deal with optical isomers the way that the tree outside is dealing with it every day, 100% of the time. It can distinguish one optical isomer from another one. It's really kind of amazing. That's where we get D and L isomers. Fischer projections. You're going to have Fischer projections, Haworth projections. There's 3 disaccharides that I want you to be aware of, and what monosaccharides make them up: sucrose, maltose, and lactose. And these are some of the key elements of this chapter. All right, what do we do with carbohydrates? We mostly use them for energy. Glucose, that is our energy supply. Some say, oh, I got low blood sugar. And what do they normally mean? My blood sugar is low. They're actually also, their energy level is low. They're sagging in terms of energy. They don't have the right amount of glucose. And it might be they're diabetic, or it might be they haven't eaten for a long time, and their glucose level in their bloodstream is dropping, and so is their energy level. So this is what we use it for. About 4 kilocalories. That's a a nutritional calorie there. 1 kilocalorie is a nutritional calorie of energy that you're getting from 1 gram of sugar. All right, I'm going to go through some of this quickly. You're going to basically find 2 functional groups. The homework asks you it asks you that. What are the 2 basic functional groups that are on every saccharide? An OH and a carbonyl. All right? Those are the 2 functional groups. Mostly OHs. There's an OH generally. I'm going to point out one key exception to that, which is kind of interesting. I think very interesting. But typically, all the saccharides you see, every carbon has an OH on it, except for one where there is a carbonyl, and that carbonyl can be an aldehyde or a ketone. And we're going to see that's one way of distinguishing, putting saccharides into 2 different categories. Is it a aldose? It's got an aldehyde functional group, or is it a ketose? Does it have a ketone as the functional group? Glucose is an aldehyde. That's what's up at the top there. That's CHO. There was a molecule like that on the test just right now. CHO is a carbon-oxygen double bond and a hydrogen. It's an aldehyde. And glucose is an aldose. All right, so another way of classifying them is as monosaccharides, disaccharides, polysaccharides. Now, this would be representing the glucose as a ring. If that molecule there were to cyclize into a ring, and we'll talk about how it does that and exactly what chemistry is involved to form the ring. But that is the way it usually appears, is as a ring. And so one ring is a monosaccharide. Here are 2 rings with a bond between them. It's a disaccharide, and that's a glycosidic bond. We'll look at what that is later, what that glycosidic bond is. Okay, so there's mono, there's di. You will see the term every so often. We won't use it a lot, but be aware of it. An oligosaccharide is a short chain of saccharides. Short, 3 to 10. Is that a hard and fast rule? No, but whenever someone's referring to an oligosaccharide, they're talking about not a polysaccharide, not many, and not 2. 2 is a disaccharide. Oligo is 3, 6, 8, 10, 12. It's an oligomer. An oligosaccharide. You'll see this prefix show up with regard to other things. You get into polymer chemistry and they'll say, no, I didn't make a polymer, I made an oligomer. What's that mean? Well, it wasn't that long. Wasn't that short, but it wasn't that long. An oligomer. Polysaccharides, many, and when we mean many, 100,000 saccharides linked together, can be a lot. They can be homopolysaccharides, they're all the same sugar. Or they can be heteropolysaccharides, there's more than one sugar. There's glucose, there's fructose, there's galactose, there's different sugars on this polysaccharide. It can be a straight chain like that, or it can have some branching. Cellulose, what makes up tree bark and stems of branches and stems of corn stalks and things are branched. They're strong. They are mechanically strong. It's kind of hard to bite into a trunk of a tree. That's cellulose. But they've got some strength to them, and how do they get that strength? From branching. Okay, now let's get into how we name saccharides. I've just mentioned a moment ago that we talk about ketoses. They've got excuse me, I'm pointing to the wrong one. Ketoses, they've got a ketone as the carbonyl. Carbonyl is not on the end, it's somewhere in the middle. It's usually 1 carbon from the top. I don't know of an example where it isn't, actually. It's usually one carbon from the top is the ketone, or they're in, so that's a ketose. This would be an aldose. The carbonyl is at the top. And when we usually draw these, we draw the carbon-oxygen double bond towards the top. For an aldehyde, it would be right at the top. For a, here's a ketose. You can see here's carbon number 1, 2, 3, 4, 5, 6. But we put that towards the top, and we're always ending down here with a CH2OH group at the bottom. So aldoses, ketoses. Another way to classify them is according to how many carbons make up the saccharide. This is 1, 2, 3, so it's a triose, 3 carbons. It's an aldose because it's got the aldehyde functionality, aldose, triose, so it's an aldotriose. None of those things are the name of that molecule. None of those, except this. Glyceraldehyde, that's the name of the molecule. These things are just kind of categorizing it. What kind of a saccharide is it? It's an aldehyde functional saccharide. It's 3 carbons long. It's glyceraldehyde. All right? Glucose, what do we got here? Glucose, it's an aldose. 1, 2, 3, 4, 5, 6. It's a hexose. So it's an aldohexose. But its name is glucose. That's the name of that molecule. And we'll talk about that. D is an important letter in front of it. And we'll talk about how we know that's D-glucose. All right, so here's another example. We've got a ketose. 1, 2, 3, 4, 5, 6 carbons long. It's a hexose, just like glucose is a hexose, but this is a ketone functional hexose. Ketohexose, that's fructose. Yes. No, we go with common names. These are common names. Yeah, we just ignore the IUPAC name of it. Yeah, this would be glucose. There's going to be about 6 or 7 of these that I'll say memorize. Let's see if I have them all here. I'll point out which ones that you should put on 3x5 cards. In a moment, I'll obviously glucose, but you don't need to write it down yet. I'll point these out. Okay? You see where we're going here? These are categories. These are names. And these are the, if you want to say, the official names of those molecules. Okay. What are these praying hands here for? You probably thought, wow, you should have put those up before the exam. Right? All right, what are they here for? Well, let me show you. Can everybody see these? Let's say we've got a carbon here, and it has 4 different things attached to it. Don't even worry about what they are. They're just all different. Maybe a hydrogen, an oxygen, who knows what else, but they're different. So I've got them colored: white, yellow, red, and green. White, yellow, red, and green. Do you think these are identical? Are these identical molecules? I'm baiting you a little bit. So if you say the wrong thing, I mean, I would look at those and they look pretty similar, don't they? These are different molecules. And let me show you. Remember back in chapter 10, 11, I said, okay, when do you know whether a molecule is really different from another molecule? I said, if you can superimpose 2 molecules on top of each other, then they don't need a separate name. They are identical. There's nothing different about them if you can superimpose them. So let's see if we can do that. I've got my carbon, so I'm going to put my red one on top of my red one, and my yellow one on top of my yellow one, and the green one is on top of the white one, and the white one is on top of the green one. So there is a difference between these two molecules. It is subtle. It is subtle. It is a difference that if I had two molecules like this, went over in the lab and said, oh, let me oxidize them, or let me do this, or let me do that, they would do those things identically. They would have the same chemistry. They got the same functional groups on them. They're not going to behave differently chemically except in a living organism. Why? Well, living organisms do chemistry better than we do it in the laboratory. And so I would like you to appreciate this. These are optical isomers. Why are the praying hands there? You look at your hands, you think, they're the same. They're not the same. Your left hand is not the same as your right hand. Your left hand is a mirror image of your right hand. It's a mirror image. You cannot superimpose your right hand on top of your left hand. These are very different. Okay? They're mirror images of one another. Okay. No, that's not why I brought this today. Okay, why did I bring this today? To show you that. How can I do this? Do they look the same? Okay. My right hand is the mirror image of my left hand. It's the mirror image of it. You guys didn't see that, did you? Right, I wanted to. You see that this hand is just here. Here I'm looking at my left hand in the mirror. You're looking at my right hand, not in the mirror, and they look the same. They're mirror images of one another. Likewise with this. Let me see how I can do this. Yeah, is that working? Does this look like the mirror image of this one? Yes? Okay. There's the mirror image of this molecule, and it looks like this molecule. Even though these two molecules cannot be superimposed upon one another, this is why we call them optical isomers. They are very, very similar. Well, there's two reasons why we call them optical isomers. One, they're mirror images of each other, and in a minute, I'm going to show you that we can distinguish one from the other in the way they behave with polarized light. So there is a behavior they have relative to light, particularly polarized light, that will distinguish this isomer from this isomer. All right, so this is optical isomers. And how do you get an optical isomer? You get optical isomers when you have four different things on the same carbon. And I'm not going to describe what I mean by different here in a minute. Let's see if that says this. Yeah, right here. A carbon atom with four different groups attached to it is called a chiral carbon. This is the main feature that creates optical isomers. If you have four different things on a carbon, then there can be an optical isomer of that molecule. All right. I should have brought glyceraldehyde in. I thought of bringing these isomers. I don't know if I brought glyceraldehyde or if I can make glyceraldehyde. I don't think I can. Yeah, glyceraldehyde. Maybe we'll pretend these two are something. Let me draw glyceraldehyde. We had it a minute ago, so glyceraldehyde is going to be, this is the simplest saccharide. It's aldehyde functional. It's got an OH, it's got an H, and then it has a C. Okay, now this bottom one right here is a CH2OH. You can see that. I mean, I could collapse these in there, CH2OH group. Now there's going to be two isomers of this. Let me draw the aldehyde exactly the same way. Okay, now to have a chiral carbon, the carbon has to have 4 different things on it. All right, so I can look at this carbon right here. Can that be a chiral carbon? Can that carbon be a chiral carbon? How many things are attached to that carbon? 1, 2, 3. Or I could call this whole thing a thing. This thing is a thing. But that carbon only has 3 things on it. Chiral carbon has to have 4 different things. So that's not a chiral carbon. That one is not. Let's look down here. Does this carbon have 4 different things attached to it? Different? No. Why? Because 2 of them are the same. It has 4 things. 1, 2, 3, 4. Has 4 things on it. But they aren't 4 different things. 2 of the things are the same. So whenever you have 2 things that are the same on the carbon, it's not a chiral carbon. So that's not chiral. But let's look at that carbon. That carbon has an OH. It has a hydrogen. It has a CH2OH group on it. And it has that. That is not the same as that. That's an aldehyde. This is a CH2OH. This is an alcohol group. Different. 4 different things. This is a chiral carbon. What does that mean? That means that glyceraldehyde comes in 2 different isomers, D and L. Let me erase this. So where'd it go? Where'd they go?
For that picture,
Yeah,
it was
more up. Oh, there it is. Okay, so when the OH group is on, if the OH group is on the left on this chiral carbon, this is the chiral carbon. So what I've got drawn here are the 2 isomers. You can see that the OH here is on the left. This is L, glyceraldehyde. And when the OH group is on the right, unfortunately it's not called R, it's called D, glyceraldehyde. Yeah, that's based off of that OH that's on the chiral carbon. That's what's determining this. And actually, glyceraldehyde ends up being our reference molecule. You're going to see how we're going to go to glucose, and we're going to look at that chiral carbon on glucose, and we're going to say, is it similar, is it the same as D-glyceraldehyde at that portion of the molecule, or L? And we're going to compare it to D and L glyceraldehyde and determine whether it's D or L glucose. Okay, now this also introduces Fischer projections. I don't know. Well, there's our oxygen. I don't. Let's say this is another OH group. This is going to introduce a Fischer projection. And what a Fischer projection is we take our molecule. I probably really need a glyceraldehyde. Let me see if I can get one. Okay, this won't take a second. So we need a carbon. We need another carbon. We need a carbonyl up at the top to make our aldehyde. Got it. We need an OH right here. This is our OH group on our glyceraldehyde. And this is going to be a hydrogen. And then down here, we're going to have two hydrogens making the glyceraldehyde. Two hydrogens. And then we're going to have an OH group. So let's see which glyceraldehyde I made. So here's what we do with this. We've got this molecule. This is glyceraldehyde. I don't even know which one I made yet. We'll figure it out right now. Fischer was a scientist looking at all these things and saying, man, we got these two isomers. We need a way of drawing these molecules. Let's see, there's a hydrogen right there. See, the model leads you. There's a hole right there, missing something. So there's our aldehyde. There's our CH2 at the bottom. And I can tell you right now that this is this isomer. Why? Because what Fischer said is, okay, here, let's develop a protocol for how to draw these molecules. And the protocol is going to be get the carbonyl at the top. Let's get the three carbons vertical in a row. And let's project the carbons behind the middle carbon. And the OH and the hydrogen is going to project outward in front of the middle carbon. You with me? So he's saying, get it in this actual position, and then imagine that you squashed it onto your paper. And what would you get? I'll draw it right there. I don't think I could squash this on my surface. Okay, so if I squash this right there, I've got, and this right here, I'm not going to even draw that carbon. I would have this here. I would have this down here. There's my CH2OH. And if I put it in that position, if I put it in that arrangement right there, squashed it on there, where would the OH end up? The OH would end up right here, and the hydrogen would end up right there. which means it would be like this isomer, so it would be the L isomer. You with me? I'm making a big deal out of this, but it is worth your while to figure out how to draw these Fisher projections. And what you have to do is line the carbons up vertically. You have to have you can't have it like this. You can't have the carbonyl underneath here. That's wrong. You look at that. It shifted the OH over to the other side. Follow me? This is the way we're looking at it. We're going to look at it with the carbonyl at the top, the CH2 at the bottom, projecting away from you, and the hydrogen and the OH are going to project towards you, and then you're going to squash it into a two-dimensional drawing. This is what he's trying to do. He said, how can we draw these on a piece of paper? Do we have to send molecules, make models, and send them back and forth in the mail? Does UPS and Federal Express have to deliver these back and forth, or can we get them into an email? Now, this was before emails. Fisher was doing all this. But how can we draw them on a piece of paper? And that's what he's getting at. Squash it onto a two-dimensional drawing, and then see where the OH ends up. And if it's on the left, it's the L isomer. If it is on the right, now notice, to get that on the right, I have to make a different molecule. I have to switch the hydrogen over here and put this over there. So there's no way to just spin around and get that over there. I can spin it like that, but now the carbons are facing you, and this is facing me. And Fisher's saying, no, you got to get it like this. Am I making sense? It's subtle, but that's a Fisher projection. Why does it matter? Nature makes D-glyceraldehyde 100% of the time. Doesn't make this isomer. It really matters when we talk about glucose. All the green plants, all the green leaves out here are producing glucose. Sunlight's shining in them, making glucose. What are they making? D-glucose. D-glucose. There is an L-glucose, but natural systems don't make it. They make D. It would take a chemist a long time, and actually, you know how a chemist would make D-glucose? Would use bacteria to make it. Wouldn't try to do it chemically. Would actually get a living organism to make it, because the living organisms can be that much more specific about its chemistry and how it does chemistry. So this is optical isomers. Glyceraldehyde is our reference molecule. And let me show you how that may pan out here. All right, so if we go over to glucose, let's make that bigger. This is glucose. We've got a carbon-oxygen double bond, an aldehyde up there. This carbon is not chiral. You know that right away. It has two hydrogens on it. It's got four things. 1, 2, 3, 4. Actually, that's the fourth thing. But two of them are the same. So this is not chiral. This one has this thing, this, and that. That is different from this. So that's a chiral carbon. This carbon has 1, 2, 3, 4. And that thing up there is different from this thing down here. So that's a chiral carbon. Likewise, that's a chiral carbon, that's a chiral carbon. And the top one is not. It only has 3 things on it. So glucose actually has 4 chiral carbons. 4 of the 6 carbons have 4 different things on them. But it's this one right here that we're going to focus on. And let me just draw glyceraldehyde right next to it. So glyceraldehyde would be this. This would be D-glyceraldehyde. What happened? Aldehyde. Something like that. All right? This is D. Why? Because this is on the right, and this is D-glucose because this is carbon number 5. It's the second one from the bottom. It's the one right up from the CH2OH group. The OH is on the right, so this is D-glucose. This is D-glyceraldehyde. This is how we're using this as our reference molecule. We're saying when this portion of the molecule resembles that portion of D-glyceraldehyde, then it's a D sugar. It's a D saccharide. Okay, now we could take this molecule and draw it real simply, just like this. We could draw it like this. And this is D-glucose. So on a Fisher projection, this is a Fisher projection. You can draw the carbons, or you don't have to draw the carbons. Where the lines are crossing is a chiral carbon. And D-glucose is going to be right, there's 6 carbons, but if you go up from the bottom, it's going to be right, right, left, right. That's D-glucose. That's where those 4 OHs need to be. Right, right, then left, right, and then you get to the aldehyde at the top. All right, D-erythrose. Look at this OH, this, all like D-glyceraldehyde. So that's D-erythrose. Okay, Fisher projections. Lines crossing through a chiral carbon represent bonds. The chiral carbon is represented by the intersection of 2 lines. The carbonyl group is placed at or near the top. At the top if it's an aldose, near the top if it's a ketose. 2 molecules facing you or out of the plane of the paper are drawn horizontally. The bonds projecting away from the plane of the paper are drawn vertically. That's the carbons. Carbons are vertically, and horizontally you're going to have the OH and the hydrogen projecting towards you. All right, so Fisher projections. This could have been like that molecule we were looking at. You squash it flat. Here is a projected drawing of it, and here it is, a 2-dimensional drawing. Just squash it down flat, and you can just have a line there representing where the carbon is, the chiral carbon. All right, so again, here's your D-glyceraldehyde, the OH is on the right. L-glyceraldehyde, the OH is on the left. Here's the Fisher projection. Here is the perspective drawings of it, where the darkened black triangles are coming out towards you, the shaded ones are fading away from you. All right, so there's D-erythros, L-erythros. D-glucose, right, right, left, right. L-glucose, left, left, right, left. All right, so this is D-glucose.
Glucose, it has the third one always has to alternate between one side. So if you had all 4 hydroxyls on one side, it would no longer be glucose.
That's correct. It's another sugar. It is another sugar, and I'll show you all of those sugars in a minute. In a minute now, we're going to shift over and only talk about D isomers of these saccharides. Why? Because that's the biologically active isomer. That's what you find in biological systems, is the D isomer. And you may not really appreciate until we get to chapter 18 how living systems can do this. But proteins are doing the work. They're doing the chemistry inside the body, and they will fit the D isomer into the protein to metabolize it, and they won't fit the L. L-glucose doesn't even taste sweet, all right? I give you L-glucose, and you say, oh, it doesn't taste like it. I don't know what it would taste like. But your body doesn't metabolize it. And it doesn't taste sweet. It doesn't activate your taste buds. It's useless. Well, it's worse than useless because it's not natural. And whether it does something messy in the body is a good question. All right, just some more slides of it. All right, let's move on. All right, let's just see. A, B, C, D, E, and F. You just tell me. Let's first go, is it an aldose or a ketose? Aldose or ketose? Ketose. Aldose. What? Ketose. Aldose. Ketose. Aldose. Okay, now I will label on here whether they're D or L. Okay, now this isn't the glyceraldehyde. Remember, glyceraldehyde has 3 carbons. This one has 4. So this is not glyceraldehyde, but is it a D or an L isomer? D. What are we going to look at? We're going to look at that carbon and say, where is the OH on that carbon? We're looking at all of these as Fisher projections, and we see the OH is on the right, which is D. B. What is B? L. Again, one up, it's on the left. C. D. D is D. That's good. D is D. What is D? What is D? What molecule is that? Glyceraldehyde. That's glyceraldehyde. And that is D-glyceraldehyde. 3 carbons. It's got an aldehyde at the top of it. There's only 1 chiral carbon, and the OH is on the right. It's D-glyceraldehyde. D. Again, we're just focusing right here. 1 carbon up from here, and that's on the right. So that's D, and that's L. Okay? You'd say, well, what about all those other H's? Don't worry about those other H's. Just focus on this chiral carbon. This one right here. And be able to distinguish a saccharide as being D or L based on the position of the OH group on that carbon. Okay, now, properties. Basically, in a laboratory, wet chemistry, if we go do chemical reactions on these saccharides, they're going to all react the same way. I could do a Benedict's test on them. I could oxidize them. They're going to react the same way. But the one distinguishing property is a physical property, and it has to do with the way they interact with polarized light. And this is where the D and the L come from. D, it rotates light, it's dextrorotatory. It rotates the light to the right, and L, levorotatory. It rotates the polarized light to the left, and you get L. This is where the letters are coming from, D and L. And what do I mean? Here's a polarimeter. Is a fairly simple device. It's got a light bulb. It has a polarized filter. The polarized filter here. So light coming out of a light bulb is rays of light, and the rays of light are oriented in all different directions. The wavelengths. The wavelength is oscillating in all different directions coming out of that light. You can pass it through a polarized filter, and it filters out all the light, most of the light, and now it has monochromatic light coming through. The wavelengths of light are all lined up. They're all waving the same way. That's polarized light. Now this is a test tube or some tube with a sugar in it. You put it down in there, the polarized light comes through, and depending upon what isomer is in that test tube, it rotates the light. So without the sample in there, you can see where the polarized light is lined up, at what angle is it lined up. You put your sample in there and you find, oh, it's gotten rotated 30 degrees to the right, 30 degrees to the left. And you can say, oh, that's the D isomer, that's the L isomer, based on its rotation of polarized light. So that is the primary difference in property, other than biological activity, which is extremely important. But that's the other thing. But in a laboratory, the way you can distinguish it basically is how it interacts with polarized light. Yeah, most compounds in natural systems are chiral. So you're going to see chiral amino acids, chiral sugars, lactic acid. D-lactic acid is in sour milk. L-lactic acid is found in living mussels. Humans metabolize D isomers of monosaccharides, but are unable to metabolize the L isomer. D-glucose tastes sweet, nutritious, important component of your diet. L-glucose tasteless and cannot be metabolized. So L-amino acids are the biologically active amino acid, and we'll look at their chirality in chapter 18. And D-amino acids are very rarely found. All right, so again, here's D-glucose. Right, right, left, right. By now you know that's one of them you got to memorize. Glucose is the most important saccharide, the most common. All right, we talked about these in an earlier slide. D-glyceride, D-glucose, D-fructose. All right, now we're really centering on D isomers. We're going to kind of ignore the L and go on and talk about D isomers only, because they're the biologically active ones. And if I skip over some of the wording on these slides, you go back and you read them and see what you're seeing there. Okay, now let's go through, and I will, on this particular slide, I will point out which saccharides you need to make 3x5 cards of. Let's start out with the trioses. 3 carbons. It's only one. Glyceraldehyde. There is no other sugar with a 3 carbon other than L-glyceraldehyde, but I just told you we're going to ignore the L isomers. So that is definitely one of your 3x5 cards. Move on up to, these are family, this is a family of D-aldoses. So we're only talking about aldehyde functional sugars here. Here's 4 carbons, erythrose and threose. And this is D-erythrose, right, right, D-threose, right, left. Okay, you wouldn't have left, left because that would be an L. If you had a left over here, that would be an L isomer. Well, we're ignoring L isomers. So you don't need to learn any of those. 5 carbons, 1, 2, 3, 4, 5 carbons long. 1, 2, 3, 4, different ones. There are 4 different monosaccharides that have 4 carbons. 5 carbons, excuse me. Okay, this one's going to be the easy one. D-ribose. Right, right, right. Ribose. They're all on the right. And you need to learn that one. You've heard of RNA. What does RNA stand for? Ribonucleic acids. Why do they call it ribo? The sugar backbone of RNA is ribose. That's the sugar backbone. Sugar phosphate backbone, but that's the sugar that makes up the RNA backbone. D-ribose. All right, so you don't need to learn these other 3, but there's a ribinose, xylose, lysose. So as you get more carbons, you get more saccharides that you can make with that many carbons. Now get up to 6 carbons. We won't go any further than that. We've already said you need to be able to draw the Fisher projection of D-glucose. Right, right, left, right. Right, right, left, right. You also need to be able to draw D-galactose. Right, left, left, right. All right, and you can see when you get up to 6 carbons, you got 2, 4, 6, 8 different monosaccharides that are aldehyde functional, but they have these hydroxyls in different positions. All on the right, 3 on the right, 1 on the left, right, right, left, right. That's your glucose. Right, right, left, left. Right, left, right, right. All right, here we go. Right, left, left, right. So different configurations, different monosaccharide. So there's 4. Make 3x5 cards of those and memorize their structure. Louder, Debbie. What?
D-glucose, D-ribose, and D-galactose. What's that one?
Glyceraldehyde. That's your 4th one. Okay, fructose, 73% sweeter than sucrose. All carbohydrates are solids at room temperature, extremely soluble in water. That should be pretty obvious to you. Back in chapter 12 when we were talking about alcohols, remember we said you get 1 OH on a molecule, it makes that molecule somewhat polar. And that polarity makes it soluble in water. What do we have here? We got a molecule that has an OH on 5 of the 6 carbons. Just about every carbon has an OH on it. I mean, this is very soluble in water. There's a lot of polarity in that molecule. And you know that. Sugars dissolve in water very readily. All right? So solubility in water because of the OH groups on all these carbons. And they allow for the sugar to be transported through the body very readily. Okay, all right, we're doing great. I don't know how great we're doing, but we're going to go on to Haworth projections. done Fisher. What's Haworth got to do with anything? Well, we just talked a minute ago that you're going to see this ring structure being used, and I should have a glucose. Someone help me. Tell you what. Put all that together. All right. Put that together. We're going to make a glucose here real quickly. We got to get a glucose because I got to cyclize one.
When we're drawing the fingerprint projections, do we need to do like the wedges and all that stuff?
No, just draw them flat lines. Yeah, the perspective formulas, it's just kind of showing you what you would do to try to draw them, but we're going to flatten them out. Got that? Well, just give me that. All right, we got those OHs? I'm not going to worry, well, maybe I will worry size of them.
No, don't put them all together. Just put them like this.
Just put it just like that, because I need these to go on here. Yeah, I just need 3 of these. That's right. Thank you. Let's put that. Come on. My leg. Maybe I am. Okay, here's glucose. Let's see if we can. All right, so what I'll do down here at the bottom, here's my here's my aldehyde at the top. This is D-glucose. 1, 2, 3, 4, 5, 6 carbons. We've got an OH on each of these carbons. Here is our CH2OH group at the bottom. Here is our bottom 3 carbons. Got the light in my eyes. Here's the bottom 3 carbons. I've got the carbons projecting away from me. This is on the right, so this would be D-glucose. This OH is on the right. Follow me? So we got D-glucose. This is carbon number 1. Now pay attention to this because these carbon numbers are going to start being important. The aldehyde functional group at the top is where this molecule starts. 1, 2, 3, 4, 5, 6. So the bottom here is carbon number 6. All right. In chapter 12, when we studied, no, in chapter 13, when we studied aldehydes, you remember this? We said that there is the hemiacetal reaction. I said would be important when we got to chapter 16. What is that? That's the reaction of an alcohol. Well, let's just put a carbon over here. We've got an alcohol reacting with an aldehyde, right? And what happens? This bond breaks, and you get that H over there. And that bond breaks, and you get a bond there. You form a carbon-oxygen-carbon bond. You form an alcohol. The carbonyl is converted to an OH. Well, what do we have in glucose? We've got a carbonyl. And what else do we have? We got alcohols all over the place. This alcohol is too close to react. You'd say, well, boy, wouldn't it be nice to be close? Well, it can't get oriented properly to add across that double bond. This one barely can. That's down 1. This one probably could do it, but this is the one that does it. carbon number 5. Carbon number 5 comes around here and can get right next to that carbonyl. You see that? We've got a carbonyl, we've got an alcohol, and we're going to get a reaction. But the carbonyl and the alcohol are on the same molecule. Okay, now let's make this react, and it's going to close a ring here. How's that going to happen? We're going to break this bond and put the hydrogen on there. That turns that one into a hydroxyl. And this is going to bond to that carbon. There's your ring. There's your ring. It's a 6-membered ring. And it has an oxygen in it. That is a cyclized glucose molecule. It has cyclized through the hemiacetal reaction. Okay, so it reacted with itself and formed a ring, closed this long chain molecule into a nice, neat little ring. And this is carbon number 1, 2, 3, 4, 5, 6. This CH2OH group is carbon number 6. All right? So now, Haworth started thinking about this. He said, man, how can we draw these? Is there some pattern that we could adopt to draw these so that we can share one molecule? I could talk about a molecule with another person. And so he came up with a way to draw these, the Haworth projection. Okay, now what is the Haworth projection? He said, okay, your oxygen is going to go up here in the upper right-hand corner of the ring. And what do I have there? This is going to be carbon number 1, 2, 3, 4, 5, and there's another carbon right up here. The CH2 is going to project above this ring, and we're going to talk about what, now there, doesn't that look like a trampoline? I'm not an artist, this is an art class. But if someone told me how to, say, draw a trampoline, I'd say, well, could I draw you alpha D glucose, and let's try to make it into a trampoline. It looks like a trampoline with a little net around it, right? There's the legs, and there's the things there for the thing, and that's what you got. Sometimes what you'll see is this kind of coming out like this and being blackened in. And what is that trying to represent? That is just trying to represent that the way I'm looking at it, I'm not looking at it like this. Okay, this is up in the right-hand corner. I'm looking at it like this, kind of looking at the ring, and then I'm asking myself, what is a below the ring and what is above the ring? All right? I should draw these down here a little bit more. All right, so this is a Haworth projection, at least the start of one. Yeah, and this is D-glucose. You have an OH group projected below the ring. You got one above the ring. The next one is below the ring. They're alternating. Below, above, below, and then the CH2OH is above. And this is D-glucose. Now that's not the full name. To get the full name now of this cyclized glucose molecule, we have to focus on that OH. Is it above or below the ring? It's above the ring, right? And that is beta D glucose. Beta D glucose. That OH in carbon number 1, the one that got formed when the carbonyl reacted with the OH on carbon number 5, is it above or below the ring? That's beta D glucose. Now you're not going to know exactly where it is unless you draw the molecule exactly this way. What do I mean by that? Exactly with the OH in this right-hand corner, with the CH2 group up above the ring. It's when you draw it that way when you draw it that way, there's my OH back there. Here's my flat ring. My CH2 is above, and I can say, oh, this oxygen and my hydrogen fell off. It's kind of not on there real well. My hydrogen is down below there. That's below. That's not beta. That's not a beta. Beta would be if this was up here. It's down there. That's alpha. Alpha D glucose. Alpha and beta. And there are both of those kinds of glucose.
But why do you base it off of that OH? Is it because it's on the first one?
It's for two reasons. It's on the first one, yes. But remember, I just took that other thing off of there. This OH was a double bonded OH. And it's reacting a double bonded oxygen. It's reacting with the OH on carbon number 5, and depending upon where this was rotated, this either ends up here or down there. So in the cyclization reaction, there are two possibilities for it to end up down below or end up above. All these other things are fixed. They're not even involved in the reaction. So they're just fixed in their position. But this is the one that has two possibilities. Depending upon where that carbonyl, that aldehyde was, when the reaction, when the ring closed, it puts this OH either below or above. This is trying to show you this right here. Here's the cyclization going on. This is carbon number 1, 2, 3, 4, 5, 6. This is the OH on carbon number 5. And if this is rotated a particular way, bam, you get the OH on the bottom, which is the alpha form. If this rotates around a different direction, this oxygen ends up above the ring, and it's beta. So you can get either alpha or beta D-glucose. And your homework assignment, one of the questions would be, I don't know, it says draw one of those. Draw alpha D-glucose. Draw the Haworth projection of alpha D-glucose. So you would have to draw this drawing. And this OH has to be below, above, below. The CH2OH has to be above. And that OH, which is carbon number 1, is below. That's alpha D-glucose. So it's only when you see alpha and beta that you're talking about the ring structure, the ring form of glucose. All right, this slide is just showing the same thing, that your glucose can come on around here and form either the alpha below, beta above. That's kind of a redundant slide. What's going on here? Well, they're ignoring the hydrogens, so they don't have the entire leg on the. In other words, you don't need to draw the hydrogens. Only draw the OHs. Don't busy this up with filling in all the hydrogens. You notice I didn't do that. We would have an OH here, an an OH here, an OH here, a CH2OH there, and I'm ignoring the hydrogens. And then if we had an OH here, this would be beta D glucose. All right, fructose can do the same thing. Don't worry too much about it. Fructose, I don't have the string of it here, but it starts out with a CH2OH. That's carbon number 1. And then the carbonyl. Then that's 2, 3, 4, 5, 6. But notice that it only forms a 5-membered ring. 1, 2, 3, 4 carbons and an oxygen. Because you've got a CH2OH and a CH2OH on both sides of this thing. But in terms of the alpha and beta and drawing Haworth projections, concentrate on glucose and knowing how to draw those and how to look at one and tell whether it is alpha or beta. Just this slide just telling you fructose also cyclizes. It forms a furan ring. Glucose forms this pyran 6-membered ring. Fructose forms a 5-membered furan ring. They're heterocyclic. Well, they're not necessarily aromatic, but they're heterocyclic. They've got carbons and oxygen and an oxygen in the ring. Okay, and here's the rules for the Haworth projection. The oxygen is always at the back. The anomeric carbon, carbon number 1, is on the right side, and the terminal CH2 group is always above the ring. Those are just some of the rules that I pointed out. Let's see, how many more slides are there? How many? A lot. Oh, we got disaccharides. Okay. Well, I'm gonna just go through the really important ones. All right. Do know the difference between glucose and galactose. Even when it cyclizes into a ring. We've already looked at glucose. This OH is below, above, below, and the CH2OH is above. Beta D galactose, below, above, above. So here is the critical difference between glucose and galactose. They're both 6 carbons long, but there is this one point here, carbon number 4, where the OH is below the ring on glucose, the OH is above the ring on galactose. Your homework asks you to draw those and point out the major difference. Just to show you, there are variations on glucose. Here's glucosamine. Well, now you know what an amine is. It's an NH2 group. You take one of these. Oh, that's galactosamine, I'm sorry. Galactosamine, there's also glucosamine, but this one's galactosamine. You take that OH off of there and replace it with an NH2, you got galactosamine. Glucosamine is, people take that as a supplement for help with their joints. Glucosamine. All right, the 3 important disaccharides. Well, let's just talk for a second about how a disaccharide forms. 2 monosaccharides condense onto one another. They eliminate a water molecule, HOH. It leaves an oxygen. So this carbon attaches to that oxygen, which is attached to that carbon, and you get a glycosidic bond right there. It's a carbon-oxygen-carbon bond. It's an ether linkage. And so if you had alpha D-glucose and beta D-glucose coming together as a disaccharide, you'd have maltose. Maltose is disaccharide made up of the two different forms of glucose. And in digestion, so if you had something that had maltose in it, you ingest it, your body digests it, it hydrolyzes it, breaks it back down into alpha and beta glucose, D-glucose. All right, here's one. Very important, sucrose. Sucrose, what do you have? You've got alpha glucose, beta fructose, D, this would be alpha D glucose, beta D fructose, condensing on one another. You've got a carbon-oxygen-carbon bond, and you've got sucrose. So sucrose is a disaccharide, glucose and fructose.
Can we make a size for that?
Can you?
Lunch parts?
No. On the homework, it will ask you what are the monosaccharides that make up sucrose, maltose, and then lactose. Lactose, it's right here. Lactose, glucose and galactose are going together to make lactose. And all right, so again, you're forming a disaccharide of these two particular monosaccharides that gives rise to two maladies or two diseases. One, in older people, they become lactose intolerant, that maybe for a long time going without milk or eating any ice cream or whatever, they lose the ability to digest lactose. But more serious problem is in infants who are unable to metabolize galactose, and you get galactosemia, the toxic buildup of galactose, because the child genetically does not have the ability to metabolize galactose. And let's see if there's anything. All right. Maybe on Wednesday we'll mention a little bit about polysaccharides. But you look over these slides on polysaccharides. That we'll go through these in 5 minutes. All right?